Uh, Thank you, Henry, for the introduction, and thank you for having me as a part of the research forum at Stevens. I'm really happy to be here and tell you all about what we've been doing in the lab, and thanks to all of those people who tuned in to know. So the lab is really focused on engineering ovarian cancer metastasis, and you know that these projects, in addition to Stevens' support, we received the support from Kaleidoscope of Hope Ovarian Cancer Foundation and Olipas, and we're really really appreciate that support. The lab uses tissue engineering and imaging approaches to understand why ovarian cancer metastasis continues during and after treatment. And I'll tell you this in a minute. Why is it so important to do this? Because even though people get heavily treated for ovarian cancer, the metastasis happens. So we don't really have a treatment for metastasis, and people die of metastasis. And we have 3 projects in the lab that focus around the metastasis field. The project 1 is we engineer organotypic cultures to study early steps of ovarian cancer metastasis. And metastasis means spread, that the cells will spread within the body. We have a second project in the lab that we engineer organotypic cultures to study late steps of ovarian cancer metastasis. And then we have a third project in the lab where we use dynamic imaging of ovarian cancer metastasis in vitro and in vivo. Why do we do this? That's the question everybody has to ask itself. We do this because 1 out of 75 women in the United States will develop ovarian cancer. Ovarian cancer originates from the fallopian tube and the ovary, and vast majority of tumors will metastasize early in the disease progression. And mortalities are extremely high as compared to other women's cancers after the diagnosis. And this actually shows you, if you compare ovarian cancer mortality rate to breast cancer, you can actually see that, you know, within a 10-year survival, about 120 months, the chances of surviving after diagnosis are very slim. It's about 15%, as opposed to breast cancer, which is close to 60%. So it's extremely important to understand why those mortality rates are so low, so high. Okay. And women die of tumors that continue to spread and obstruct vital organs in spite of heavy chemotherapy, which I mentioned this already earlier. So what are 2 questions? We have really 2 questions in the lab that we try to answer. And the first question is, what drives ovarian cancer spread and metastasis? And second is, why do new metastases continue to evolve after chemotherapy? And both questions are extremely clinically important for development of new therapeutics or maintenance strategies to maintain low disease burden. So now I'm going to tell you a little bit about the ovarian cancer metastasis, so you know what I'm going to be talking about in the next few slides. So this is the little animation that we've prepared a while ago to sort of focus the audience where ovarian cancer initiates and what it does. So it initiates from the fallopian tube or the ovary. Both are the sites of origin. And the unique peculiar characteristic of these tumors is that these tumors can grow and detach. And as you can see, they populate a peritoneal cavity. And this is a massive metastasis, massive dissemination within the peritoneal cavity. And those tumors that can survive this metastasis will basically attach to the additional organs. And this could be a small bowel wall. And that attachment will cause obstruction of those organs. So the most frequent death of ovarian cancer patient is an obstruction of a wall of the small bowel. Okay, so the first project, it's what drives ovarian cancer metastasis. And this project really took off when undergraduate student Douglas Shang and Antonia Pavlovich graduate student came in the lab, and then they started to think about, where do we start to study ovarian cancer metastasis? And we need to go back to origin. And as I mentioned to you, majority of ovarian tumors originate from the fallopian tube, and this is the laparoscopic picture of the fallopian tube that encapsulates the ovary. And then if you look at in the tissue section, so basically what you've done, you took a piece of that fallopian tube tissue, you section it and you stain it to visualize cells. You can actually see that, in the normal situation, the tissue of the fallopian tube is nice and thin. This is what we call benign tumor when you have some proliferation, but it's not metastatic yet. And those tumors are rarely detected, almost never. And then you have a third situation when you have a metastatic tumors, when they make these outgrowths, what we call them in the lab outgrowths. Those outgrowths can detach, just like I showed you on this movie, and populate peritoneal cavity. And then the phenomenal thing that we can do is we can take those outgrowths from patients, and then we can implant them in the mouse. And in the mouse host, these outgrowths will repopulate and will form the same kind of outgrowths. Now, the mouse experiment is very expensive and it requires a lot of mice to get some statistical analysis. That's why we sort of turn into a tissue engineering approach to mimic this process. How do we get from the benign tumor to the metastatic tumor? And this is the picture that shows you that we took those fallopian tube cells that are normal and the fallopian tube cells that are metastatic, malignant, or sort of those. And we were able to engineer the environment around those cells that they could make those outgrowths. So we thought that we had in hand a tool that we could study how the ovarian, drives ovarian cancer metastasis. And again, I'm going to show you a movie showing you how those outgrowths are actually formed. So at the upper panel, upper three movies are the normal fallopian tube tissue, and the lower movies is this malignant fallopian tube tissue, and you can immediately see how different they are. And what is really beautiful here is that they can recapitulate the clinical outcome. They can recapitulate what we see in clinics. So once we optimize these culture conditions, we've engineered those culture conditions, we really wondered whether we can put different ovarian cancer tumors isolated from patients, and whether we can actually observe this outgrowth formation, this metastatic outgrowth formation. And the answer is yes. So with the 3 graduate students in the lab, they screened the whole panel of ovarian cancer cell lines and looked for those outgrowths. And we saw in multiple of those cell lines outgrowth formation, and this is quantified on the graph right here. And the most important thing, the cells that are normal don't form those outgrowths. So again, we think we have a nice model to study what drives the ovarian cancer metastasis. So that's the project 1. Now, the project 2, we really want to focus on the late step of metastasis. So after you've made the outgrowth and you've detached, what are those mechanisms? And if you can see this is the peritoneal cavity of the patient. Again, laparoscopic picture, where it's seeded with plenty of metastasis right here. Okay, so those are when I showed you on the movies, those are the chunks of tumors that were dispersed in the peritoneal cavity, and now they sort of populated the peritoneal wall right here. Now, again, if you cut the tissue out of it, what you can see the tumor is laying on the wall of bowel, but it made a hole in this a part of the tissue which we call mesothelial cells. So we were really interested how we can mimic this, how we can engineer outside of the patient's body to mimic this situation for studying to understand how these processes are driven. So we obtained mesothelial cells, we obtained the tumors, and we cultured them and made the movies. We used the imaging to see sort of dynamics of this tumor and mesothelial cell interactions. And you can actually see from this movie that the moment the tumors touch the mesothelial cells, they literally make a hole in the mesothelial cells, just like here in the space and in the tissue space in the patient. And the beauty of this model is that we can actually quantify it over time, the ability of making that hole. So graduate student Sadaf Farsinajat in the lab, she was really interested in the question, well, we have these now organotypic cultures or organoids that we can recapitulate the outgrowth formation. Here's in the red, and she called this outgrowth body or the spheroid body. And then she asked the question, well, what's those things that give the rise to metastatic tumors, are they better in making those holes, or are they better in cleaning mesothelial cells? And the answer was yes. She observed that on the average, these outgrowths were much better in penetrating mesothelial cells. So now we have a model that we can actually study the mechanism and try to log that process. Okay, so project number 3 is that we are really, really interested in understanding why during the chemotherapy and after chemotherapy, this is a patient data, why those metastases continue to evolve, to continue to happen. And Tonya Pavlovich and Sadaf again tried to mimic the patient's situation when they took those engineered organotypic cultures and they treated them with the chemotherapy, recovered them, and asked the questions, are there any different after the recovery? And there was a striking difference. We observed that the organoids that recovered chemotherapy, they would make more outgrowths, and they would be pretty rough on the outside. And this is sort of a quantification of that phenotype that Tony has done, that the chemotherapy actually promotes this sort of fragmented situation when the tumors can metastasize. And this is quantified right here as well. And what Sadaf's recent experiment suggested that if she stimulates acutely with the chemotherapy, this mesothelial clearance is inhibited. However, if she recovers the cells after chemotherapy, mimicking the patient situation, because in patients you receive the chemotherapy and then you recover for 1 week, these tumors completely regain their ability to clear the mesothelium. So we are really understanding what drives this regaining ability to clear the mesothelium. Okay, now we understand, and you know, I'm very aware that studying cancer just in vitro, basically in vitro, sort of we're missing on a lot of factors, and it's a mainly microenvironment. So we really are looking for the in vivo approaches, especially that we were able to recapitulate the outgrowths in vivo. And we collaborate with Shang Wang on sort of developing some of these in vivo approaches to track the metastasis in vivo. So Shang beautifully developed this intravital window that you can actually track you know, contraction of the fallopian tube or the ovary. You can track the sperm, and it's a beautiful tool for us to actually ask the questions, can this happen in vivo? And we're particularly excited about this project because we received a fundable score from National Cancer Institute, and we are really hopeful that this is going to be funded. So with all of that, I'd like to, you know, this wouldn't be possible. This is probably the most important slide on this presentation. This wouldn't be possible from all these without all these people in the lab, undergraduate students, Dak and Estefan, high school student, Fernanda, who spent the summer really trying to grow those organoids, and of course, my graduate students, Sadaf, Sarah, Daniel, and Tonya. And I'd like to also acknowledge close collaborations with you know, people from biomedical engineering and CCB, and those are all the people that we've written grants together, and we're hoping to have this supported also through the interdisciplinary approach through the funding. Thank you so much, and I will take gladly any questions.
Yeah, thanks very much, Marcin. This is a critically important research area. Greatly appreciate your efforts. So questions from the audience. Once again, please unmute yourself and also identify yourself before asking any questions.
Can I ask?
Yes.
Hello.
Yeah,
yes, I can.
Yes.
Yeah.
Alexander Sutin, it's Sensor Technology Center. So my question is, did you try to apply ultrasound imaging, for example, ultrasound microscopy for ovarian? Because in this case, ultrasound can penetrate and to detect something without vision, direct vision.
Yes, so we haven't, so this is again, you know, specialty of Shang Wang. We haven't tried ultrasound, but Shang is really interested in using optical coherence tomography to look at those lesions and how those lesions develop in a mouse. Now, as far as detection, because I think your question also sort of leans into detection strategies, that people do intravaginal ultrasounds to detect whether you see any lesions on the fallopian tube or the ovary. Unfortunately, those fallopian tube lesions are really small, so you won't be able to see them by ultrasound. You've got to have a big tumor to actually start seeing this by ultrasound. The short answer to your question is we haven't tried the ultrasound. Yeah.
Are you interested to try?
That would be fantastic if we can come up with, in the mouse model, if we come up with the strategy to actually use the ultrasound to follow the dynamics of these metastatic processes. Yes, I mean, that's just learning experience, of course. Yes.
Okay, we can discuss this option.
Yes, thank you so much for your question. Any more
Any
questions?
other questions from the audience? Yeah, if not, Marcin, I have a question. So regarding early detection of ovarian cancer, right?
Yes.
Are there any biomarkers that may be present in the blood or even urine of?
Yes. So unfortunately, there are not, and there's a biomarker called CA125, which is a blood biomarker, but that biomarker doesn't really detect early. It usually is a late indication of ovarian cancer. And the problem also is that different people have a different levels just from the start of that biomarker, right? Your background levels is different. So there are not. So, you know, the fallopian tube origin, which I mentioned at the beginning of the presentation, is a relatively new paradigm in the field, because people thought for last, 75 years that this was coming out of the ovary, but as a matter of fact, it turns out that it comes out of the fallopian tube. So now growing these fallopian tubes now and as an organoid or having mouse models will definitely support the development of those biomarkers. But again, short answer is no. There's no biomarkers for early detection. That's why it's so deadly.
Yes, indeed. It's certainly depressing to see that. So we look forward to a breakthrough development in your research lab to counter such a critical and yet deadly challenges that women face.
Yeah. So
okay.
before I go, I just want to say that September is the Ovarian Cancer Awareness Month, and so if you have a chance, please wear your green ribbon. It's to support ovarian cancer research and the patients, the most important. Thank you, Henry. Thank you, everyone.